Hello, you're watching a Unipro UGENE podcast. Here we discuss the hottest topics introduced by our users and show some helpful tips for using UGENE, a free cross-platform genome analysis suite. A codon table was added to the sequence view. It depends on the translation code selected for the sequence. To show or hide the table, use the Ctrl+T shortcut or click the button on the toolbar. Clicking on a codon name redirects you to Wikipedia to give you a brief description of the corresponding amino acid. Cells of the table are colored according to classes of amino acids. UGENE now supports annotation of amino acid sequences. It performs annotation in 2 steps using the Uniprot BLAST and Distributed Annotation System, Protocol 1.6. Open the DAS Annotations tab on the Options panel for an amino acid sequence. First, you search for orthologs of the current sequence, sending a remote request to the Uniprot BLAST server. Notice you can configure the database to search in and the similarity identity. Let's find orthologs of the sample sequence by clicking the Fetch IDs button. The IDs of orthologs are shown in the table. Then, using the selected IDs, UGENE sends requests to DAS sources to upload annotations by the IDs. To do that, we need to select IDs and click the Fetch Annotations button. The annotations are added to the sequence view of the current sequence. By obtaining annotations of orthologs, UGENE helps one to understand functional properties of the regions of your amino acid sequences. The Workflow Designer version of the algorithm can be used for batching annotations. Another new feature available in UGENE 1.13 is the advanced NCBI Genbank search interface. It allows you to easily search for DNA and protein sequences in the Genbank database. The interface can be accessed from the main menu, File, Search NCBI Genbank. As you can see, the visual interface is similar to the one which is used on the Genbank webpage. So if you've used it before, then you won't have any problem searching for a sequence from UGENE. To search for the sequences, just type your query into the term field. For example, let's search for gene KPNB1. We will type the gene name into the field. One can customize the search by providing additional terms. This can be done by clicking the plus button. Let's further customize the search and filter out only for human sequences. This can be done by selecting Filter Organism and typing Human into the term field. Notice how the resulting search query is modified while you add new terms. It is possible to search both for nucleotide and protein sequences by selecting the corresponding database. Once the term is constructed, just click Search or press the Enter button. The result of the search will look like a table. Each row represents one sequence or annotation. To download the sequence, just select the items in the table and click Download. More than one item can be selected at a time. In the Download dialog, it is possible to set the output folder for the sequence file. After clicking OK, the download will start in the background, and one can continue the search or close the dialog and start working with the sequences. A major new feature of the UGENE Workflow Designer is the Common Workflow Output Directory. If you open the Workflow Designer for the first time, UGENE allows you to choose a directory where all information about your workflows is stored. If you choose to use the directory, UGENE will create a separate subdirectory of the Workflow Output Directory for each workflow run. Such a subdirectory stores files produced during the workflow run, the dashboard associated with the run, and even the workflow itself, with all the parameters filled. When you set the output file for your workflow, you do not specify the path to the real file, but you use a special dialog that helps you to specify the relative path in the subdirectory of the run. You can create directories with the dialog and use the helpful extension suggestions. After running the workflow, you can see the results in the dashboard. You can run the workflow again, and the results will be stored in another subdirectory. If you close a dashboard, it is not fully deleted. You can restore it with the Dashboards Manager. With the help of the Dashboards Manager, you can open, close, and remove dashboards. Dashboards are useful for managing your workflows and monitoring their results. You can see the output and input data of the workflow, time consumption, and logs of external processes. In the new version of UGENE, the ability to analyze the workflow process in runtime was added to the Workflow Designer. This feature is disabled by default, but you can switch it on in the application settings. From now on, you can investigate the process of workflow execution. Let's consider a workflow sample for this purpose. This workflow performs a search of specified patterns in each sequence of the input file and annotates found regions. Here we can create a breakpoint. It may be thought of as a particular mark on a workflow element that forces the workflow to pause when the specified element starts processing incoming messages. The pause is a new workflow state besides existing running and idle states. This mode allows you to observe and analyze the workflow intermediate data. Let's see it in action. Now the workflow is paused, and you can choose any message queue in it and observe the intermediate data stored in the queue. Here you see the instrument called Investigator. It shows a summary of each message in the workflow channel. The main part of the Investigator is represented by the table whose rows match messages in the queue and whose columns match different pieces of data added to the particular message by the corresponding workflow element. For instance, you can see the column named Read Sequence Sequence. That means that the column represents biological sequences written to workflow messages by the Read Sequence element. If you are interested about a particular message's content, you can save it into a file with an appropriate format and then open it in UGENE. Eventually, you can analyze this sequence the same way as an ordinary sequence. This approach of obtaining the workflow's intermediate data may be utilized to obtain data of all the types supported by UGENE. Such data analysis may be useful for long-running workflows when you are not sure if you have set all the parameters correctly and the workflow will actually produce the results you expect. In this case, you may check intermediate data on the first workflow steps and either stop the workflow or resume it depending on your examination of the result. Another tool for the debug mode in the Workflow Designer is Breakpoint Manager. It contains a list of breakpoints existing for the current workflow. Yellow highlighting specifies the breakpoint that has triggered last. Here you can specify the hit count required for the breakpoint to trigger. Besides just specifying the hit count, there are some additional parameters available. For instance, you can make the breakpoint trigger, say, each 5th time. Now let's resume the workflow. And we see that it has paused after 5 messages passed through the find substrings element. To sum up, the described debug means introduced in this version of UGENE may turn out quite useful in analyzing sophisticated and time-consuming workflows. There are even more debug features available in the Workflow Designer, and you may find them in our documentation. The new version of UGENE is able to recognize PHYLIP format and open it in the alignment editor. Both sequential and interleaved forms of PHYLIP format are supported. UGENE detects which form is used automatically, but you can choose the preferable form manually. Also, in case of doubt, UGENE suggests the appropriate format to use. ACE files are now imported into the UGENE database files and opened in the assembly browser. By default, the file is created in the same location as the input ACE file. You can see the resulting contigs aligned to the reference sequence. The Bowtie 2 tool for aligning sequencing reads to a long reference sequence was added to UGENE. It is integrated as an external tool. To align the reads using Bowtie 2, select this method in the dialog and input files with the reference sequence and with the reads. When aligning paired reads with Bowtie 2, set the library spin box to paired end and specify the mates order. Set additional options if required and click Start to start the aligning. The resulting SAM file can be imported to the UGENE database and displayed in the assembly browser.